Good afternoon. My name is Seoyeon from the TAF School, and today I'll be presenting my research on investigating whether oxytocin can protect human brain cells from cortisol-induced damage, specifically by preserving oxytocin receptor and BDNF expression. Adolescents show heightened cortisol reactivity at precisely the developmental stage when peer relationships and emotional regulation become the most important. This creates kind of a paradox where the brain is most vulnerable to stress hormones exactly when social bonding matters the most. I hypothesized that oxytocin mitigates cortisol-induced stress in human brain cells, that cortisol would suppress OXTR and BDNF expression while oxytocin co-treatment would restore them. For the experiment, I used human glial A172 cells across 4 groups. Control, cortisol only at 1 micromolar, oxytocin only at 3 micromolar, and co-treatment. After 24 hours, I assessed OXTR and BDNF expression via immunofluorescence and validated with RT-qPCR results. Here are the dose-response selection of working concentrations. Presta Blue viability assays confirmed that 1 micromolar cortisol and 3 micromolar oxytocin were subcytotoxic, maintaining approximately 90 and 95% viability respectively. Cortisol-treated cells showed visible cytoskeletal collapse. They had flattened morphology with retracted processes compared to healthy, elongated, and extended controls. Immunofluorescence revealed cortisol reduced OXTR to 65% of control and BDNF expression levels to 85%, both statistically significant. Oxytocin alone, on the other hand, produced a 4-fold increase in OXTR and a 1.6-fold increase in BDNF, suggesting a positive feedback mechanism. Here was the key finding, where during the co-treatment, oxytocin did not merely reverse the suppression, it amplified OXTR expression to over 3.5 times the cortisol-only level, even surpassing the untreated controls despite ongoing stress. RT-qPCR results confirmed these effects at the transcriptional level. Both OXTR and BDNF mRNA mirrored the protein level patterns across all 4 conditions. I propose that cortisol activates glucocorticoid receptors to express OXTR and BDNF transcription, while oxytocin binding triggers GQ-MAP kinase cascades that restore expression and establish a self-amplifying feedback loop. These findings carry implications for adolescents' mental health, but are limited by the use of glioblastoma-derived cell line, a single time point, and small replication number. Future work should employ primary cells and also chronic exposure models. This study demonstrates that oxytocin may act as a molecular shield against cortisol-induced damage. Social support may not be merely a psychological comfort, but is rather a biological necessity. Thank you.